Hello, and welcome everyone. In this podcast, we're going to talk about chromosomal mutations that involve changing the chromosome number. So, chromosome number changes. In this regard, we're going to talk about anything that deviates from what the euploid is. The euploid, EU, stands for true, so this is what the true ploidy of that organism is. And just as an easy reference, we're going to think about humans in these examples here. And so humans, we have a diploid number of chromosomes, which is equal to 46. Now that can deviate in the number of chromosomes by either an aneuploid, which is 2n minus or plus 1 chromosome. I write 1, but that can actually be different. It doesn't always have to be 1. Another example would be polyploids. And these are when you have a whole extra set of chromosomes. So it'd be like going from a 2n to a 3n, a diploid to a triploid. And for humans, that would be going from 46 chromosomes to 69 chromosomes. Or you could imagine it's a diploid going into a tetraploid. And again, that would be 46 going into 92. Now we're going to come back to the polyploids later in this podcast. But for now, let's go ahead and erase this. And let's focus on the aneuploids. And remember, I said it could be plus or minus 1, and then I said there doesn't always have to be 1. It could be plus or minus 2, 3, but not the whole set. The whole set would be a polyploid then. So let's talk about some terms here related to the aneuploids. You could have a nullosomy, and this would be 2n, that diploid number, minus 2 chromosomes. So in humans, that would give us only 44 chromosomes. We also have something called a monosomy. And this would be 2n minus 1 chromosome. In this case here, we would have 45 chromosomes in a human. We could also have a trisomy. And this would be a 2n plus 1 chromosome. In this case, we would have 47 chromosomes. 46 chromosomes for our diploid plus 1 more for 47. And finally, we could have a tetrasomy. These would be the most common deviations from the euploid of 46 chromosomes. Tetrasomy would be 2n plus 2 chromosomes. So that would give us 48 chromosomes. As you're preparing for the exam, a question I often like to ask is giving you an organism other than humans. Say I tell you there's this organism that has 54 chromosomes. I'm going to want you to be able to tell me how many chromosomes there are in that organism that is monosomy, trisomy, or tetrasomy. So don't just remember these numbers, remember how we get those numbers. For the most part, in our discussions for aneuploids in this podcast, we're going to focus on monosomies and trisomies. So a question we might have is, why are these a problem? Well, let's look at it this way. Let's draw a chromosome here. And this would be the euploid. And they would have, from these 2 chromosomes, their expression would be 100%. Meaning the expression that you would need for that organism to survive is 100%. In the case of a monosomy, where you're going to lose that chromosome, I'm just going to put a dashed line around here just to remind us that indeed there should be something there, but it's not there. In this case, we're only getting 50% of our expression. So this is a monosomy. Alternatively, we could have a trisomy, where we have 3 copies of this chromosome. And in this situation here, we're going to have 150% of the expression of genes. So that's 50% more than just the standard euploid. And again, that was a trisomy. So this is why we have altered phenotypes, and in most cases quite severe phenotypes. Remember, each of these chromosomes may contain hundreds of genes all the way to thousands of genes. And if you think of this chromosome containing, say, 1,000 genes, if we automatically upregulate 1,000 genes by 50%, you're bound to have some genes on that chromosome that will not tolerate that. Sometimes genes can tolerate being in threefold here, but it's rare to find a chromosome that's going to tolerate that and allow for a viable birth. The next question I want to ask is, how do they form? So how do monosomies and trisomy, how do these form? We can come up with 3 different ways that would facilitate these monosomies or trisomies from forming. The first is a centromere deletion. So remember, if you have 2 chromosomes, homologous chromosomes here, and the region of this that gets deleted is the area around this one chromosome. As a result, this chromosome will no longer have a centromere. If it's missing a centromere, you're not going to be able to bind to it during anaphase and mitosis or meiosis. As a result, this chromosome here without the centromere will be lost. So this one is lost, and this one survives. Unfortunately, most monosomies in this case either sick or it's lethal. They'll die as well. Monosomies are just this one class chromosomal mutations that is not tolerated very well in animals, including mammals. The other example here is the result from a Robertsonian translocation. So remember, this is a situation where you have 2 acrocentric chromosomes. And these acrocentric chromosomes are non-homologous. So let's make this one a little longer. And they can combine by a mechanism that we talked about on the last podcast, but they can combine and form one metacentric chromosome, as we've said before. So we're going to get the long tail on the blue one and the long tail on the black one. And then we'll get this short puny tail from the black chromosome and then from the blue as well. Often, as we've said before, this smaller chromosome that forms from these small tails here essentially gets lost. Sometimes they persist for a little while, but usually they don't last for a very long time. So this is how we form that inheritable form of Down syndrome, if you recall. And when you have that, then that can also lead then to trisomies. The last way we can generate chromosomal changes, aneuploids that is through something called non-disjunction. We're not going to spend much time on it here, but in the last podcast, we're going to go pretty in depth in what non-disjunction means. But for now, what you need to know is that homologous chromosomes, or in some cases sister chromatids, stick to each other. So either homologous chromosomes or sister chromatids, they're supposed to stay separated, but every now and then we can get them sticking with each other. And when that happens, it can lead to a non-disjunction. We'll come back to non-disjunctions later though. So how often do these aneuploids occur? Well, remember that we have a germline, before it's gone through meiosis, with 46 chromosomes. And that gets broken into 23 gametes forming. Now sometimes we have a deviation in these. Maybe we only have 22 in this one. Or maybe you have 24 chromosomes in that one. So when this combines with a gamete that has 23 chromosomes, we're only going to have a total of 45. Here we're going to have a total of 47 if they were to meet a euploid gamete. So how often do these kind of errors occur? They occur in approximately 5 to 10% of fertilized eggs. So in about 5 to 10% of the cases, the fertilized egg or the zygote will have one of these mutations. So this is a pretty high number, 5 to 10%. So what happens to those aneuploids? Well, most are spontaneously aborted. Scientists will then study these aborted fetuses to make some predictions about fetal development. Even though about 5 to 10% of the fertilized eggs result in a spontaneous abortion, we do know that, this is a pretty big number, 50% of all spontaneous abortions are caused by chromosome changes. So how do we know that these always occur? Well, we know that these occur. In fact, we know that they occur with most chromosomes. And we know this by looking at the tissues of aborted fetuses. So I'm going to move away from the whiteboard for a moment and show you a karyotype. So we talked about how common aneuploids were and how often they led to spontaneous abortions. But as we look at this karyotype here, we can see we have 2 copies of every chromosome. 203, 204, 205, and all the way down here to having 2 copies of the sex chromosomes. If a trisomy were to occur, we would have an extra copy here. So the question I want to ask is, which of these chromosomes allow trisomies to form? Which ones of these in humans are monosomies allowed to form? Which one of these can tolerate having 1 whole extra chromosome? And which ones can tolerate having one of those chromosomes removed? Now we know that trisomies can occur in every one of these chromosomes, with a possible question mark by chromosome 1. However, only a few of them can survive to birth. These include chromosome 13. If it's a trisomy, it can survive to birth, so we have an extra one here. Chromosome 18 can survive as a trisomy. And then chromosome 21, right here, can survive as a trisomy. Now in each of these cases, 13, 18, and 21, and I'm going to list them on the whiteboard in just a moment, but all of these result in a birth. However, most of these don't make it to birth. Most will be aborted spontaneously. Again, all of these can form trisomies, but they all lead to spontaneous abortions. Now, the only ones that can tolerate a trisomy or a monosomy and survive, and the progeny has a fairly healthy future, would be trisomies and monosomies of the X chromosomes. So, for instance, you could have an extra X here with the Y, so it'd be 2X and a Y. We'll talk about that in a moment. You could also have a situation where you have 2 Ys and a single X. You could also have a situation where you have 3 X's and no Ys. And we'll give all 3 of those conditions a name in just a moment. But usually the sex chromosome aneuploids not only survive, but are fairly healthy. So question is, why do most of these lead to a spontaneous abortion? And why do some not? Okay, let's move back to the whiteboard. Okay, so I want to list right now the viable aneuploids. So these are aneuploids. I'm going to break that up down into trisomies and then monosomies. Remember, trisomies are going to have a total of 47 chromosomes. Monosomies are going to have a total of 45 chromosomes. Well, of the trisomies, we can have chromosome 13 exists as a trisomy. This has the name of Patau syndrome, P-A-T-A-U. We can also have a viable trisomy 18. This is known as Edwards syndrome. We can have a viable 21, which is Down syndrome. I should mention that these individuals that have Patau syndrome, Edwards syndrome, or Down syndrome, if they survive to birth, they typically suffer from many anomalies, which is in sharp contrast to the sex chromosome trisomies. We can have XXX, which we just call XXX, no Y chromosome, and we'll learn that the Y chromosome is really just this puny little chromosome that has very few genes on it. We could also have an XXY. These individuals, we call them Klinefelters. We can also have an one X with two Ys. This is called Jacob's syndrome. These typically, there are phenotypes associated with them, very few with the XXX, but Klinefelter, these are male, and they typically are very tall. They are also have some more feminine phenotypes associated with them, and they are sterile. Jacob's syndrome, there are some traits linked to them, sometimes increased acne or increased ADHD. At one time, they thought they had a propensity towards violence linked to the two Ys, but the studies that showed that were very flawed in the sense that they only looked at people who were in prison, so it might have been a logical assumption that you were going to have people with a propensity towards violence regardless of their chromosomes here. Alright, now, monosomies. There are no autosome monosomies. With the sex chromosomes, there's only one that's tolerated. XO. There's not really an O chromosome, that's just meaning there's just one X chromosome there. And we call this Turner syndrome. Not having a Y chromosome, these individuals are female. They are often sterile. They're often shorter in stature. But other than that, you wouldn't know they have this syndrome. One of the leading researchers on Turner syndrome, she herself has Turner syndrome. Now again, I want to stress that these are the only ones we see in live births, but we know that the others occur. We know that other trisomies at other chromosomes occur, and other monosomies at other chromosomes occur. They just don't make it to birth. And we know this by studying spontaneous aborted fetuses. And again, I should also stress that all of these, including the sex chromosome aneuploids, most of them don't survive to birth either. So are there any risks associated with an increase in aneuploid children? There are a few, not a lot. We'll mention two here. One is if you happen to be a Robertsonian carrier, like the 1421 we discussed when we talked about that special case with inherited form of Down syndrome. So that would be a risk factor. Another risk is the age of the parent. This is primarily associated with maternal age, but not exclusively. We know as males age that there is an increased risk of having a child with an aneuploid. However, it's at a pretty high age range, typically at an age that is beyond when most men have children. Maternal age. This seems to be a bigger factor. And we know that by age 40, the risk of an aneuploid goes up quite a bit. We know that the overall risk of something like Down syndrome, let's focus on Down syndrome here, is about 1 in every 800 births. So it's pretty common. However, this risk of 1 in 800 isn't the same at every age. I'm going to erase this just so we have a little bit more room to show you this graph. Here we have maternal age. And over here we have Down syndrome incidence. And I'm going to draw this line like so. We know at about age 20 here, there's like a 1 in 1900 chance. we know at age 40, that goes up quite a bit to about 1 in 100. At age 45, it's about 1 in 32 chances. And by age 50, that number goes way up to about 1 in 12. So we have this really drastic, really slow climb, but then once you hit age 40, the incidence of Down syndrome that is provided from the egg goes up dramatically. So the question now is, why? And we'll come back to this in class. Why does it go up? Again, we don't see this increase with paternal age until the father gets in their 70s or 80s. Then we start seeing the number go up. In fact, in all cases of Down syndrome, only about 5% come from the sperm. That is, only about 5% of the sperm contain an extra copy of chromosome 21. Next, I want to talk about polyploids. Now, polyploids exist when you have a whole extra set of your chromosomes. So when you go from a diploid to a triploid, or a diploid to a tetraploid. I'd like to take a step back and look at this picture of a karyotype from humans that is a polyploid. That it's actually a triploid. And you can see here, this was taken from a spontaneously aborted fetus, and you can see that we have 3 copies of each of the chromosomes, all the way down to the X, where we have 3 copies of the X. Now again, these are lethal, and these individuals don't survive, but we can recover the tissue from spontaneously aborted fetuses and be able to tell why they didn't survive. In fact, it's very common to do this because when you have some couples who routinely have or have had several spontaneous abortions, it's good to know why. And if you can pinpoint it to something like this, that helps the genetic counselors understand how to treat the couple and make suggestions to them. Okay, let's go back to the whiteboard. Alright, I'd like to now just mention some observations about polyploids and what kind of organisms we see them in. Now, mammals, I'm going to put very rare. And some would say never. There's only one species of a rat that's been found in Argentina that some believed was a polyploid. Others don't believe it was a polyploid, so it's a little debatable. So I think for our purposes, we'll just go with very rare, if at all. Now, birds, it's not known, or not observed, I should say. We've never seen it. It might exist, but we just haven't seen a polyploid bird. Now, when we start looking at organisms like fish, amphibians, and reptiles, it's more common. Not overly common, but you do see them in fish, amphibians, and reptiles. I know of a few examples in amphibians where frogs exist as tetraploids, 4 copies of each chromosome. Now, where we see a big jump in polyploids is when we look at our plants. In plants, and we'll talk about a couple of examples here, in flowering plants, we know there's about 40% of them that are polyploid. In grasses, we know about 70% are polyploids. So again, they're very well tolerated in plants. And this benefits them, helps them survive better, and humans, as we cultivate plants and breed plants, we've taken advantage of the fact that they're polyploids. And we'll talk about that in a moment. Now, we said that polyploids don't usually exist in animals. We do see some things that are close to polyploids in animals that bear mentioning here. And the first is sometimes animals are what is called haplodiploid. So not a 3N or 4N, but in this it's a special case where one gender, and it's usually the males, it doesn't have to be, but it usually is, the males are haploids, and the females are diploid. It could be vice versa, but usually it is the male that's haploid. This is very common in ants, bees, wasps, where the males are haploids and the females are diploid. Another interesting example that we can see in humans is something called Polyploid. And this is when a tissue or organ has polyploid cells. And a great example of this is the human liver. In the human liver, we find all sorts of chromosomal mutations, extra copies, either aneuploids or polyploids, chromosomal rearrangements, but it works well in the liver for its purposes. And I want to come back to plants. And we already talked about how many plants, flowering plants, grasses, ferns, are polyploids. But some features of these polyploids are that they are usually larger than their non-polyploid relatives. And the reason for this is that they have, remember we talked about G1, S, G2, yes, we're coming back to it, sorry, and M phase. The reason polyploids are a little bit bigger has a lot to do with the fact that when their cells grow, they have extended G1s stages and to some extent G2s stages. So that's why they're a little bit bigger. Now this is something that helps the plants themselves, and that is they tend to be a little bit more stress tolerant. Now this other one, it's hard to imagine this is much of an advantage to the plant, but it's an advantage to us, and that is they tend to be sterile. And the reason that's an advantage to us is that's how we get seedless fruit. We know that many of the fruits and vegetables that we buy are polyploids. For instance, just to throw a couple out at you here, cotton, is a tetraploid, has 4 copies. Oats have 6 copies. Potatoes that you buy at the store, they're tetraploids, they have 4 copies. Strawberries, they're octoploid, they have 8 copies. Same thing with sugarcane. They tend to be larger than their counterparts, more tolerant to stress, and they tend to be sterile. And the reason they're sterile is, again, we're going to go back to our good friend here, meiosis. And let's think about just one set of these chromosomes, and we're going to talk about a triploid. And we're going to draw the chromosomes over here. We're going to have one set of sister chromatids in blue, one set in orange, and one set in purple. And when these line up along the metaphase plate, in fact, I'm going to move this circle over. When these line up here and begin to separate, you could imagine a situation where the orange and the blue here pair together, are kind of stuck together, and then this purple one is by itself here. And when this divides, and you make 2 new cells here, you have your blue one here, and your orange one here, at the end of meiosis 1, that is. your purple one here. Now the purple one here, it's going to separate out, and you're going to have gametes with one of the sister chromatids in one of the gametes and the other sister chromatid in the other gamete. So you might think, well, that's going to be fine. This is going to be a gamete that will be viable. Well, the problem is we only talked about one of these chromosomes. Remember, these are polyploids. So if they have, and plants tend to have a lot of chromosomes, but let's just say they had 20 chromosomes. The chances that you're going to get one gamete formed where each of the chromosomes segregate just this way, so that this one gamete right here will have one copy of each chromosome, is exceedingly rare. It's not impossible. Every now and then one of these sterile plants can produce a seed, but it's very, very rare. And the way I drew this here was just one possibility. You could have a possibility like this, where they all kind of stick together and synapse with each other. And when that happens, all three end up in this cell, and nothing in this one. So again, with so many chromosomes, the chances that you're going to get one gamete that's produced that has just one copy of the chromosome in it is very rare, and usually isn't going to happen. Okay, that's all I have to say about aneuploids and polyploids, but let's give a quick summary. Remember, we talked about aneuploids. You should know the difference between a euploid, an aneuploid that is either a trisomy or a monosomy. You should know the different viable aneuploids, both the autosomes and the sex chromosomes. You should know why they don't survive or do as well. And again, it goes back to the fact that you have so many more genes overexpressed at one time. You should know a little bit about the current. How often is this? And some of the risk factors. And we talked about the main one. Well, two, we talked about the Robertsonian for the inherited form, translocations here, or age, and how that was a bigger factor for women. We also talked about polyploids. We talked about which kind of organisms they form in. We talked about the special cases of the haplodiploid and the endopolyploid. Remember, that is when you have tissue that has polyploid chromosomes in them, even though the rest of the organism is diploid. And we spent some time talking about plants and why it's so important for some plants to be polyploid. Alright, if you have any questions about this material, please make sure you come see me. If not, I'll see you in class. Bye.